All right, welcome back. And this will be the second lecture and also the third lecture of the pro-seminar Decision Neuroscience. So I changed the order slightly. So second lecture will be about a refresher of experimental design and statistical inference. And then the third one will be about research methods used in decision neuroscience. So from today on, I also would like to get to know something called a decision warm-up, decision neuroscience warm-up. It's basically to ask ourselves what we have discussed from the last session. Doesn't make too much sense today because the last session, last week, we only talked about the introduction and how we will be graded. I'll still take a few moments to think about what we have talked about decision making. So in the restaurant example, if we are interested in eating out regularly, a few months ago, for example, we search on the Google map to say maybe Italian restaurants, and Google will return to us a few choice options, right? So choice options, a few options, decision options to choose from. This is one property. And also recall the example where I asked you to choose from a chocolate brownie versus a cucumber smoothie. Which one you chose from really depends on your internal goal. For example, if you just want to treat yourself, maybe chocolate brownie, or in the current situation, you do not want to gain too much weight, then in this case, a healthy greenish smoothie might be the choice for many of you. Okay, so those are the things we talked about, not too many. We will cover that in the next session of today. But before that, so as psychology students from the bachelor program in the University of Vienna, so I'm sure that most of you have taken some kind of courses or contents regarding the two topics. One is experimental design and the other one is statistical inference. So I think why this can be important, it is because we, at the very end of this seminar, we will have a group project, as I discussed the last time. We will have a experimental design exercise or project as a group, so that you propose one experiment looking at specific parts of decision making. But before that, we need to understand what experimental design are and what can be used to do statistical analysis. So the purpose of this session is mainly to serve as a refresher. So I said I'm sure that most of you know this already, but I just want to quickly recap what you know already. Maybe something you don't know, I will just also quickly run through about that, okay? Good. So from the survey I showed you the last time, so many of you have already experienced a behavioral experiment from the faculty, maybe even fMRI experiment, EEG experiment from the faculty. So it might be that you have a rough understanding of how research can be conducted. So I'm now presenting you something called the research cycle. So usually it starts from here. You're interested in one question. You specify, you formulate a hypothesis, and then you design an experiment to test that hypothesis. And because you design an experiment, you want to collect data to prove or maybe to falsify your hypothesis. So how to do that? You have to collect real data. So here you move down to collect some real data. So this is the running experimental part, right? So if you have participated in any of the experimental experiments, then you are basically contributing to the data of the project, which is important, very important. Next, as a researcher, when we have data collected, we need to analyze the data. We need to make statistical inference. For example, if we want to compare if there is any difference between two groups, and then we basically have to run the appropriate test to test if there is a difference or not. And after we have the statistical results, by analyzing the data, we need to interpret the data to see if our results are proving or falsifying our hypothesis. And then we publish our experiments, and then maybe we run a new study. And then this cycle goes on and on. So this is the quite abstract idea of how research could work, not only in psychology. So this can also be applied to some other fields. So biology, for example, it will follow the same principle and the same steps. So the first part of this section, this lecture will be about experimental design. So you might wonder, so why on earth do we need experimental design? So we are all curious about mental processes and behavior. For example, we might wonder, why do many people distrust the government? We might also wonder, why do many people hoard toilet paper during social crisis, for instance. People do that. People are interested in this behavior. We as psychologists, we are also interested in this kind of behavior, this kind of questions. So what is the difference? So what distinguishes psychology researchers from the others? So the question is answered by here. So the method the psychology researchers use to produce and develop their knowledge. Okay, this is the difference in between people who are doing psychology research and people who are not doing psychology research. The scientific methods. Usually there are two stages when you are using this kind of scientific methods. Both of the two stages, they are already described in the research cycle. So one is to formulate hypotheses. So what might underlie a phenomenon? And then you want to test your hypothesis. What are the evidence? Is that supporting or does that falsify the hypothesis? So usually you might wonder how to formulate or how to specify a hypothesis. The first thing is actually you do not have a hypothesis at the very beginning. What you have at the very beginning is a research idea, right? So here, we want to show how some ideas can be translated or formulated into hypotheses. So here are a few examples. So young captive monkeys who are treated nicely and receive clear signs of affection from their caretakers are likely to develop into nice, sociable individuals. So this is one idea, right? Second example, so if we choose something without any reward from choosing it, we will have a stronger sense that we have chosen something we really like. And the third idea is that the more we focus on the meaning of sentences, the better we remember them. Okay? So those are the ideas. Maybe the idea comes from your observation, your own experience of learning, of observing the others, from your parents, from your friends. So those are ideas from you, right? Doesn't matter where they come from. Or maybe from literature. If you are doing some kind of research, you read paper, and from your bachelor study, you read paper, and you might find some study interesting, and you want to go further from it, and you might have ideas. Those are like ideas. And then think about a minute, maybe 10 seconds. How, for example, this one. How to translate or how to specify a testable hypothesis from the idea. Yeah, just think about a few seconds. So because I asked you this one, then let's look at here. So what a hypothesis could look like in response to this idea. So let's say here, sentences for which people are asked to think of an adjective that summarizes their meaning will be more easily recalled than sentences for which people are asked to count their syllables. Just one type of hypothesis. And if we go here, this hypothesis is that if people spontaneously choose one thing over the other, they will be more confident that their choice was a good one than if they had received a financial reward for making that choice. Here, this hypothesis for the first research idea is that, okay, so daily touching and stroking captive monkeys in the first 6 months of their life will increase the number of interactions in which they get involved between 7 and 36 months of age. So you see the difference a little bit. So the idea is that for a research idea, those are not very specific and maybe a little bit vague. Just idea that you want, that is a question you are curious about, that is something you want to know the answer. But to know the answer, you might have to be a little bit more specific. So here, you see first 6 months of their life, and then here, between 7 and 36 months of their age. This is quite specific, right? And then here, there's some comparison. If you want to answer if this is true or not, you have to be a little bit specific. In one condition, in one scenario, people just choose spontaneously. And then in another scenario, there is a financial reward involved. And then here, related to this research idea, the corresponding hypothesis is about, okay, which part of the sentence they might have to focus on. So one is the adjective, the other is only like the syllables of the sentence. Okay, so those are just the hypotheses coming from research ideas. So here is a kind of a short summary. So how to formulate hypotheses? It can be based on existing theory, reading papers. It can also be from your personal experience, okay? So now you can basically ask yourself some kind of the question like, here in this category, research idea, you can ask yourself, for example, social isolation will reduce people's general motivation regarding food, regarding money, for example. This is one idea, right? It's kind of timely. And then you might want to hypothesize, to formulate a hypothesis, to test that hypothesis. Then what can be specific, for example, is that social isolation of one day, the outcome, the behavior of seeking food is different from being social isolated for 2 weeks. So then there is a comparison. So maybe socially isolating one person for 1 day is okay for human because they can still eat, and then they have, in particular, if they have internet, they can still be connected with the outer world by using Facebook, for example. But for 10 days, even with this food, with this social networks, with social media, it's quite challenging. So then that might be some motivation reduction. So it's simple ideas, simple hypothesis to test, okay? And then after hypotheses are formulated, the next job of a researcher is to test the hypothesis by designing experiments. So that's the purpose of the session, of the lecture. So to test the hypothesis, different methods can be used, such as, for example, systematic observation or laboratory experiments. So we will then talk about different methods, in particular, experiments. So experiments in psychology, they constitute a very powerful technique for the investigation of causal links between different things. And this is why they're ideal for testing causal hypotheses. So it is a master plan that specifies the methods and procedures for collecting and analyzing the needed information or needed data in a study. So one key important feature of experiments is that experiments are well controlled. So researchers here, they manipulate a variable. For example, let's call it X, and then keep the other variables constant. So then in this case, any difference in the observed observation, in the observed data, the Y, could only be attributed to the difference in X. So this is why running experiments could be used for testing causal hypotheses. If, let's say, we run one observational study, we observe two variables, and if, let's say, they are positively correlated, is there a causal effect or not? You can then ask yourself, is there a causal effect or not? Is that only a correlation, or will there be some confounding variables? You don't know, right? It is not well controlled. So in experiments, we have two conditions: social isolation for 1 day, social isolation for 10 days. Let's say we keep all of the other the same. We only look at people within the range of between 20 and 30 years old. And we look at, for example, educational level, at least finished high school study. And we look at, for example, only healthy people without any without any psychological disorders, psychiatric disorders. So we control any aspect. The only difference between the two groups is that one is being isolated for 1 day and the other one is being isolated for 10 days. And then we look at how they differ in the behavior of interest that we want to test. Okay? So one other example experiment I can give you is here, this one. So we have a hypothesis. We could say we are interested in this question. The more positive the mood of people, positive, negative, similar to the isolation example. So the more positive the mood of the people, the better their intellectual performance. So for example, if they're happy, if I ask them to do an IQ test, so their performance might be higher if they are neutral, or maybe if then they are unhappy, right? So this is the hypothesis we are interested in. And then to test this one, to test this hypothesis, we need to do some experiments. So now what I want to do is that I show you how this example works, and then later I will explain different elements in this experiment. And then through that example, we will see how experiments might look like. So let's say, as usual, 40 just healthy individuals are selected for participation in the experiment here. And once they arrive in the lab, participants are asked to take part in some activities. Actually, unknown to the participants, they're randomly split into two groups. So one half of the 20 participants, they will watch some funny events, some videos, for example, funny events. And then the other group here, they will only watch ordinary events, neutral, boring events. Maybe not boring, but just neutral. And then both of the groups, they will complete a general reasoning test based on 10 logical problems, similar to an IQ test. And then the number of problems correctly solved by each participant is counted. So there are 10 questions. If they answer all the 10 questions correctly, their accuracy is 100%. If they only answer 7 questions correctly, then this accuracy will be 70%, right? And then what can be done is then to compare the accuracy from this group versus the accuracy from this group. Okay, this idea makes sense. This is the experiment. Good. Any questions? 5 seconds for you to ask some questions. Okay, if not, so let's talk about the elements, element by element within this experiment. The first element is called variables. What are variables in experiments? So in one experiment, the things that are expected to change are known as variables. So basically, they're not fixed. If, for example, we ask a participant, let's say 10 participants, to finish an IQ test, it can't be possible that all of them have the same accuracy. It's not possible that all of them, they have answered 9 questions correctly out of 10. So it's changing. So that's because it's varying from some ranges, then it is called a variable. Okay, the things they are expected to change. It is not fixed. The variable in my experiment that is manipulated and whose change are supposed to produce changes on another variable is called an independent variable. So short for IV, independent variable. Sometimes it's also called stimulus or just something called X for independent variables. So now what is the independent variable in our example? So what is manipulated? So what is manipulated is here, the grouping. So we have 40 individuals, and then half of them is manipulated to watch funny events, and another half of the participants is manipulated to watch neutral events. They don't know, right? We as a researcher, we make this manipulation, we make this grouping, and then this variable is manipulated. So this grouping here, this versus this, is a independent variable. Independent variable has levels. For example, if they watch funny events, they might be in this positive emotion mood. If they watch ordinary events, they might be then in the neutral mood, right? Here there are 2 levels. It can also be 3 levels. So if there is a drug test, so let's say there is a true drug, there is a placebo, there is a control, they don't take anything. So here just 3 conditions are the 3 levels of this independent variable. And in decision making, we could give participants some options to choose from. For example, we could let them choose risky options, 2 risky options. One, the risk is higher than the other. So 2 options. The risk of one option is higher than the risk of another. So they have to choose between 2 risky options. And another scenario might be that one option is safe, another one is risky, to see how they behave. So these are another example of the levels of independent variable. So next, the variable whose levels depends on the levels of a prior variable is defined as a dependent variable, short for DV or response or Y. Again, we could go back to the example. So what is the dependent variable? You can also think about it. Basically, what is measured? What is the response if we want to use the term response variable? The response is here, right? So participants, they complete IQ logical problem tests. So the response is how often they complete the questions correctly. So what the accuracy is. So this is the response. And then putting this framework, independent variable and dependent variable, relative to our previous example, we can say, okay, independent variable here listed, dependent variable here listed. We have one experimental condition, they watch funny events. We have control condition, ordinary events. And then the dependent variable is what is measured, so the response from the participants, as I said. Any questions regarding the variables, dependent, independent? Everything's all right? Okay. So the next very important aspect of experiments is called manipulation checks. So you might heard about that before. So usually if we are in the class, I might just ask you, raise your hand if you have already heard about the term called manipulation checks. You can say to me in your mind, yes or no. The purpose of manipulation checks in this example is to tell us, so how do we know watching funny versus not funny videos induces positive versus neutral moods, right? So recall the question. The research question is about positive mood will induce more higher performance on intellectual tests. So mood to intellectual performance. It's not about watching funny events will improve the intellectual performance, right? This is different. So how do we know watching the funny events will get us to a positive mood? So this is the question. So basically, how do you know the experiment works? If you ask them to watch funny versus non-funny videos, if they don't really differ in their moods, then this experiment is failed. So you see the difference now, right? So how to check in this particular example is that we could ask participants to define their mood by specifying whether it is good or neutral or bad, or maybe also to give a rating on a scale between 1 to 9. So the idea is that if we ask them to watch the video, and immediately after they finish their watching, we ask them to rate their mood on a 1 to 9 scale. So then we compare the group of people who are in this funny condition, their rating, if it is higher than the rating from the other neutral group. And if this is the case, we can say the experiment is successful. The experimental manipulation is successful. There's one very important aspect to pay attention to. It is that the manipulation check has to be orthogonal to the dependent variable. So it has to be independent of the dependent variable. So they have to be different. So we use the manipulation check to make sure the experiment induces the conditions we want, but not to check whether the experiment disconfirms or conforms of our hypothesis. So let's say we want to do manipulation check. We want to check if the IQ performance of the funny group is higher than the IQ performance of the non-funny condition. Is this a manipulation check? So this is not a manipulation check because this already directly answers your research question. This is not to check if your experimental manipulation is successful. Does that make sense? Or any questions? So only when we are sure that watching funny videos relative to non-funny videos will induce positive relative to neutral mood, we can say that comparing the IQ performance between the two groups makes more sense. There's a question asks, can I say again why it is not a manipulation check? So comparing, let me go back. So this is the experiment. Our hypothesis is more positive modes will lead to higher intellectual performance. So this is the experiment, right? If we do this experiment, we have two groups. Here they watch funny events, here they watch ordinary events. If we compare the IQ performance of this group versus the IQ performance of this group, then this already answers the question, but we still don't know if the experimental manipulation works. We still don't know if the funny events will induce the positive mood or not. So they are different things. So manipulation checks here, what we have in this example is that we ask them, how do you feel after watching this? How do you feel after watching this? So maybe, supposedly, the mood from this group is higher than the mood of this group. So then we are sure that we are manipulating their mood. And then we are sure that the mood of this group is more positive than this. And then we can answer the question, how this improved or enhanced mood is impacting the intellectual behavior, intellectual performance on the response variable. Make sense? Yes, good. Any other questions? Unclear? I think this is quite important. So when you are reading papers from, let's say, psychology in general, usually there is a short paragraph. So they say, the section is called manipulation checks. So they select some of the variable, which is not the response, as I said. Same as here. So they check some variable, which is not the response, which is relative to the manipulation. They want to check if the manipulation is successful. And only when the manipulation is successful, they can go forward. Otherwise, the experiment is too confounded. Yeah, there is a question. So before conducting the IQ test, you need to do a scale of their mood in order to do the manipulation check. Yes. Yeah, we do here. Participants come to the lab, randomly being allocated to each of the two groups. They watch the events, and immediately after that, we give them the scales, the ratings. How happy are you, or how do you feel? How do you feel? And then they give the rating. And then we use the rating to do manipulation check. And then here we go forward to let them perform the IQ test. There is a question, it says, so how do we know it is not the different events, the different events the two groups are watching that is influencing their intellectual performance rather than their mood? Then this is your question, right? This is a research question. So the question here, I think, is that if they watch here, what is changed, is not their mood, is their IQ. So then this requires a strong link, right? This requires a strong theoretical background to say, well, people watch funny videos, they will have a higher IQ performance. Let's say up to this point, we don't know. And we do this experiment to answer this question. But why we use funny videos to induce positive mood? This might be something already established. So we know from, for example, previous literature, we say, if we want to manipulate participants' emotional mood, we show them different videos. And there's no direct link to say, watching different kind of video will have different IQ score. There's no direct link. But here, that is the research question. Does that make sense? Okay, good. So let's move on. So recall from the example. The example here, it says that participants are randomly split into two groups. One, they watch funny videos. The other one, they do not. They watch ordinary. What does this randomly mean? So what does randomization mean? So here, the key idea is randomization. So if I say, I have 40 participants, the first one go here, the second one go here, third go here, the fourth go here. So it's always odd number, they go to this group, even number, they go to this group. Is that a good idea or not? So why we want to do some randomization? It is because ideally, in a very ideal scenario, we have here independent variable mood states, right? We are interested in if this one is affecting intellectual performance, the response variable, the dependent variable. This is the ideal case, then everything is well controlled. But we all know that people are quite complex, humans are complex. There are things that cannot be well controlled. For example, if one participant is tested in the morning, the other one is tested in the afternoon, and then also the environment, the room temperature is different, the noise level is different, and then the third level is also different. Maybe one participant, they run 10 kilometers before coming to the experiment. This is different, right? This might affect their performance in the experiment. Anxiety, for example, also maybe they are the baseline IQ performance. We have no idea. So it will be very difficult to control all of them. So the very first one might be easy. So we can say we only test participants 11 o'clock in the morning. This is okay. We schedule all of the participants at 10 o'clock in the morning. And maybe we tell them to not do any exercise before you come to the lab. Let's assume they're honest. They really do not do anything before coming to the lab. So there's something we can control, but there are still some other aspects we cannot really fully control. For example, we can't really say we only recruit participants whose IQ score is between 100 and 110, 120, 110. This is a bit difficult. Their anxiety level, room temperature, also not something very easy to control. So how to deal with those nuisance variables, or also sometimes called confounding variables. So what to do here is to randomize them. So let's say we randomly sample a person from the randomly sample a participant from a population, and then we randomly sample another one. So if we randomly sample different participants from the population, we might imagine that the average of their intelligence, their IQ score will be pretty much similar to the average of the overall population. So some people, their IQ is high, some people, their IQ is low. That's all right. And if we recruit more than enough participants, the IQ score will be averaged out. So then there will be a balance. It's okay. It is okay that some people, they have high IQ, some people, they have low IQ. Then eventually it will be more or less similar to the overall average of the population. And then here, for example, the temperature aspects, maybe on some day the room temperature is high, maybe on some day the room temperature is low. This is nature, we cannot control. But eventually, if we run more than enough participants, the different averaged temperature will be just even. There will be even. So on average, there is no impact. There is no systematic change from temperature. There is no systematic change from here, the participants' baseline intelligence, okay? So this is the importance of randomization. So a simple randomization procedure in this example, it could be that whenever there is a participant, they come into the lab. So what we do is that we flip a coin each time a new participant arrives, and allocating the participant to the funny condition if the coin is a head, or to the ordinary condition if a tail appears. 50-50 chance, randomly, just randomly. Good. So the purpose, again, let me just repeat. The purpose of randomization is to control for nuisance variables, or sometimes called confounding variables. For instance, here, something like here, we cannot fully control. So a short exercise for you, so pay attention. The question is this experiment appropriately designed? So let me read for you. The project students want to examine how caffeine affects performance on the memory test. All participants were asked not to consume caffeine drinks within 24 hours before the test. The first 20 participants to come to the department were given a cup of caffeine coffee before the memory test. The next 20 participants to come to the lab were given a cup of decaffeinated coffee before the memory test. The test performance of each group was used as dependent variable. Okay, so this is description of the experiment. Is that appropriately designed? To make it easier, I turned that into a multiple choice question. So what is wrong with this experimental design? A, nothing is wrong, it's perfect. B, allocating of participants to the groups. C, sample size. Okay, just write in the chat box, A, B, or C. Yeah, perfect. Yeah. So far everyone just wrote B, which is the correct answer to this question. So if I have 40 participants in total, the first 20, they come to the first condition, the second 20 come to the second condition, then it is totally not randomized. It is actually very systematic. So in this case, we cannot really get everything well controlled. Yeah, there's a question. Also, it would be good to evaluate the average regular caffeine intake before, right? Because this might affect people differently. Yes, in general, yes. But here, the experimenter, they asked the participants not to drink any caffeine drinks 24 hours before the test, so that maybe everything is back to the baseline. So might be. But recording how often they drink caffeine drinks, it is a very good strategy to have a regular monitoring of how much caffeine they intake every day. Yeah, good point. Thanks. There's another question. They would also have withdrawal symptoms. Yes, might be, I guess. So as a serious experimenter, if some people, they do have withdrawal symptoms, then this has to be taken into consideration during the experiments. So let's say we have randomly assigned 20 people to the caffeine group, right? And then if they have some symptoms, and then maybe those people will be left out for the analysis, and then you might want to have a few more participants in this group. It might be one solution, but maybe there are smarter solutions. There's another question. They don't know who comes when, so isn't it randomized? I don't understand the question. Maybe can you elaborate? You can talk over your microphone. Anna? You.
Hello?
Yeah, I can hear you. Yes, go ahead.
Hear me? So they don't know which participants comes into the room. And if they give the first 20 participants the caffeine, isn't it randomized because they don't know who comes in?
Yes,
you know
okay.
what I mean?
Yes. So here, why it is not a good idea, it is because maybe you see these advertisements of recruiting participants. Maybe the first 20 people, they are more motivated. So then they are already very interested in this study. And then the next 20 people here, they're not very interested. So there's a confounding variable, which is the motivation. So here, if there's any difference on the memory test, might not be caffeine, it might be motivation. If they're more motivated, they want to come to the experiments, then their IQ, not IQ, their memory test might be higher than the memory test performance of the second group of people who might be less motivated. You see what I mean? So if there's any difference on memory test here and here, it might not be because of caffeine, it might be because motivation. The first 20 people, they're more motivated in taking the experiment relative to the second group of people. So then to get rid of this to get rid of this motivation confounding variable, so then to randomly allocate each person, maybe same idea, coin flipping 50-50, to allocate each person to each testing group, caffeine or decaffeined. The next topic is about type of experiments. So if you read some textbook, for example, there are something called true experiments and also something called quasi-experiments. So true experiments means involve full control of the experimenter over allocation and scheduling of treatments. So it is a complete randomization. For example, this placebo control or caffeine, decaffeine, just randomly allocates each participant to each of the two groups. So quasi-experiment means not full control over the allocation of participants to the different levels of the independent variables. So this is a partial randomization. For example, if we want to study if there is any difference, learning performance difference between autistic patients versus healthy control. We can't say, well, random participants, we randomly let the participant develop autistic symptoms. This is not ethical, right? We can't do this. We just use this natural group. Some people, they might be diagnosed autism. Some people, they're healthy. We use this naturally forming groups as a variable. But you see, this is not a full control. So this is a partial control, a partial randomization. In this case, quasi-experiments. And also, you might heard something called a within subject design. So within subject design is that suppose we have multiple conditions, multiple levels of one independent variable. So the participant is tested under all the conditions. So for instance, we could be interested in the learning performance at the beginning versus at the end of the semester. So same person, for example, I give you 10 questions. Now at the beginning of the semester, you give me the answer. After our learning, I give you the same 10 questions, maybe different 10 questions, but relatively overlapping content. I give you another 10 questions, and then I ask you the question, and then I compare your performance at the end of the semester versus at the beginning of the semester. So you are the same person, you are tested twice, okay? So within design. So suppose there's a healthy population, for example, in this case, we randomly sample a few people here and here. So actually there shouldn't be a line here, there shouldn't be a space. So all of the people, they will be treated using treatment number 1 and also treatment number 2. So the treatment here is just a generic term for condition. It doesn't have to be treatment, medical treatment or something like that. So treatment 1 can be beginning of the semester, treatment 2 can be the end of the semester, okay? So all of the participants, they are tested under all the conditions. So within subject design. By contrast, another type is called between subject design. So by the name, you can already imagine. Each participant is tested in only one condition. For example, in our IQ, mood state to IQ example, so the performance here in this example, the mood, so one group of people, they only watch funny events. The other group of people, they only watch the ordinary events. They do not watch both. Okay, so this is a between subject design. So suppose this is the population, we sample some 2 groups. So one group, they take the treatment 1, the other group, they take the treatment 2, okay? This is one case. So in another case, for the quasi-experiment we talked about, so let's assume there are autistic people here shown this color, light yellow. There are this darker yellow color. They are just, for example, healthy control people. So here, then this the illustration of the quasi-experiment. Group 1 is like the naturally healthy control. Group 2 is the naturally formed autistic individuals. And then this is also a between subject design. There is a question, so shouldn't the mood prior to the experiment also be considered? Yes, very good point. Yes, so in your way, it is a very interesting point. All right, yes. So here, 40 individuals, here they randomly got allocated. So before they watch the video, there can also be a question, how do you feel, right? How do you feel before watching, and then how do you feel after watching? And then here, this rating after watching can be used as a manipulation check. And for example, if this experiment is very successful, we then do not expect any difference in the rating before they watch each of the videos. So how do you feel here should not be so different from how do you feel here before watching. And then after that, we will see, okay, maybe the induced positive mood is only because they watch the funny events, right? So then this makes this experiment more technically sound. Make sense? Okay, good. We talked about within design and between design. And both of, oops, sorry. So both this design, the within design, and here this between design, we only have two levels of independent variable. So here treatment 1, treatment 2. Here you can also say this treatment 1, treatment 2. So autistic individuals versus control. Here treatment 1, treatment 2. So we only have two levels of the independent variable. But what if we have multiple independent variables? Multiple levels of the independent variable. The corresponding design is called factorial design. So I can give you one example here. So we have factor A. So again, it is a video watching kind of example. So factor A is the content on TV. Is that violence? Does that contain violence or not? Okay. Level 1, yes, some violent content. No, non-violent content. Factor B is something different. It is realism. So the first level is that do the characters in these videos, are they real? Yes, they are real. No, they are just cartoons. So then we have a two level here, sorry, I use this. Then we have a two level here of factor A. We then have two level here of factor B. Then we could have 4 different combinations. So if you read papers, if you see there is a 2x2 experimental design, that usually means this. 2 levels from factor A and 2 levels from factor B. So the combination is obvious. So here, violent, real, violent, cartoon, non-violent, real, non-violent, cartoon, okay? So those are 4 here, different cells. And each cell means one combination between the levels from the 2 factors. So suppose we run this experiment, we can recruit participants. We can say, let's run a full within subject participant. So each participant, they will watch video from all these conditions, all 4 conditions, okay? So supposedly, we ask all the participants to watch all those videos. In this case, it is called also a repeated measures factorial design, because all those people are repeatedly measured in different experimental conditions. So this is the idea. So we have here, for instance, 12 participants, 1, 2, 3, 4, and there are 4 experimental conditions. So as you can imagine here, so the question here is it's a good idea to always show the videos in this order. In this order means real violence first, and then they watch real non-violence, and then they watch cartoon violence, and then they watch cartoon non-violence. So it's a good idea to watch the videos always in this order. So relative to the randomization, this is another place you can also randomize. So you do not randomize the group allocation here because this is a within design. All of the participants, they will have to undergo all those experimental conditions. But what is randomized is the order of how they will be presented in terms of showing the stimuli, right? So here, what I talked about is a potential nuisance or confounding variable, which is the ordering effect. So it is not ideal to always have this order. Instead, we have to disturb the order of showing each content of the cartoon or each content of the video, not always cartoon, sorry. So the possible solution, if you read paper, this word might also be familiar to you. So it is called counterbalance. So let's say there are 4 different types of order. So here they watch this condition first, and then the second, and the third, and fourth. So here maybe some other participants, they watch the second condition first, and then third, and fourth, and first. And here, so always shift a little bit, so that on average, the order is counterbalanced. So some people, they watch this one first, some people, they watch this first, some people, they watch this first, and some people, they watch this first. Similar idea to this IQ, right? So if some people, they have high IQ, fine. Some people, they have low IQ, also fine. On average, this will be even, this will be balanced. Similar here, we cannot really say within one person how this order will affect the performance. We are more interested in the group performance as a whole. So on average, if we test more than enough participants, the ordering, it will be washed out, it will be balanced, it will be even by showing this order, by doing this counterbalanced order. So if you recruit participants, let's say, we could have the first 10 people, the first 3 participants, they use this order. And then the second group of 3 participants, 4 to 6, they do this order, and then so on and so forth. So in this way, overall, the order from the stimuli is counterbalanced. Another type of design is called the mixed factorial design. What does mixed mean? A mixed design means there is both a within subject component, there is also a within subject component. So both between and within, So in this example, we could say, well, we have 16 participants in total. The first 8, they only watch violent videos, but they watch both the real characters and also the cartoon. So here, this is the within part. But here, that's the between part. So the second part of the participants, they are only watching these non-violent videos, both here real and cartoon. There is a question. So the question is about why does it stay in the same order just with a different starting point? So why is always 1, 2, 3, 4? You always shift a little bit. Can't you do 3, 4, 2, 1, for example, right? That's the question. Yes, so we know that there are 1, 2, 3, 4 stimuli. If you want to do a full randomization, you can ask yourself how many combinations there are. There are quite a lot, right? It's 24, I guess. So anyway, it's many, it's a lot. And if you have only 24 participants, you have 24 orders. You want to have all of them experience at least one order, that's okay. So if you have 24 combinations, you have 24 participants, then all of them at least experience one type of order. That's totally fine. But for example, if you only have 12 participants, so how can you make sure all of them experience the full range of the combination? It's not possible. So the solution, why it's called counterbalance, it is not called the full randomization. So it is to take a subset of combinations. So we keep the general order, it always goes up from the small number, go to the up. This order is the same. And then we shift it so that it saves a lot of logistic issues. When we were recruiting participants, this is a lot easier than doing full randomization. Yeah, guess that makes sense. Good. Okay, so here this is the mixed factorial design. This is the between part, and then here there's the within part. Okay, this is again a 2x2 design. So if you read literature, if you read paper, if you see 2x2 design, you don't really know what that is, right? It might also be a both between design. So usually if they write paper, they always will specify which is a within factor and which is a between factor. So pay attention to that. So there are other types of study design, not full experiments, not really well controlled. So for example, the first one is correlational research. Correlation research, we are interested in the relationship between, for example, stress level and physical symptoms. So what we do is basically we measure their stress level, we measure their physical symptoms, and we do a simple correlation. Pay attention here. So correlation never implies causation. So why is that? So you always know, I guess, this famous example. So in summer, for instance, there is a high correlation between ice cream consumption and robbery rates, for example. So let's say the data shows there is a strong correlation between these two, but does that mean eating ice cream will increase your chance to rob somebody, right? This is not really true. So it might be that the temperature is the cause. So when the temperature is high, people are more likely to buy ice cream. And also in another case, when the temperature is high, people might be more likely to rob somebody. So then what you observe is only this part, but the actual underlying cause is this. You always have to pay attention to that. So correlation does not imply causation. So another type of study design is survey research. So for example, I could ask you a few questions regarding empathy. So how do you feel when you watch somebody else is in pain? So this is the question study. You do that very often. And I guess in the current situation, there are quite a lot of questionnaires going on, and all of them, they belong to this category, survey research. So the third example is observational research. For example, we are interested in how infants naturally behave within their first 3 to 6 months. So there is a baby, we just watch, and then maybe we take videos, and then we analyze the video. So when do they start walking, and when do they start laughing, when do they start calling mom, mama, something like that. Just natural observation. We do not do any manipulation. Yeah, and so on and so forth. So those are the types of study design. Good, any questions? Good. So, okay, so give me 10 more minutes and then I'll finish, we can have a break. So we talk about study design, right? So the next step, the next part of this session is also to talk about statistical inference. So the statistical inference is related to this part in the research cycle to analyze the data. So the general idea is that if we have some effects, let's say there is a cause, and then we could do experiments. So the effect is being caused by this reason, okay? So this is the cause, this is the experiment. But it might often be that this cause is unknown, it's not observable. So we want to use the data to infer what might be the cause. So from the data to this cause here, this step is called statistical inference. So statistical inference, back again to our example here, 20 people here watching funny events, 20 people here watching ordinary events, and then we test their IQ performance. So what we do is that we compare if the IQ performance is the same or different between this group and this group. So I'm sure you know this from your statistics class. There are two types of hypotheses. So statistical hypothesis. So one is the null hypothesis, H0. There is no difference in performance between the conditions. Alternative hypothesis, sometimes called H1 or HE, means experimental hypothesis. This says there is a difference in performance between the conditions. So if you understand the concept of p-value, I will talk to you later. So if you understand the concept of p-value, assuming the null hypothesis is true, so what is the probability to observe this data? Is that extreme or is that not? Okay, and then according to this probability, you will use that to either reject the null hypothesis or maybe just fail to reject the hypothesis. Okay, let's first talk about the testing framework. So this entire statistical testing framework, it is called null hypothesis significance testing. So this NHST is a method of statistical inference by which an experimental factor is tested against a hypothesis of no effect or no relationship based on a given observation. There is something called the p-value, which is the threshold, which is the alpha in your statistical analysis. So the p-value is used to reject the null hypothesis. So what is the p-value on Earth? We have, again, a test question. So type your response in the chat box. I will read it for you now. So the p-value, A, the probability of failing to reject the null hypothesis given the observed results. B, the probability that the null hypothesis is true given the observed results. C, the probability of observing results as extreme as or more extreme than currently observed, given that the null hypothesis is true. D, the probability that the observed results are statistically significant, given that the null hypothesis is true. So A, B, or C, or D, write it down in the chat box, please. All right, so even though that's not all of you have given your response, so almost all of you have correctly spotted the correct answer, which is C. So let me repeat again. So the p-value, it is the probability of observing results as extreme or more extreme than currently observed, given that the null hypothesis is true. Okay. if that is still vague, we could look at this curve. So for example, assuming there is an underlying normal distribution under the null hypothesis, in most of the case, we will observe some data, there is no difference. So remember that null hypothesis means there's no difference. So if here we assume that the null hypothesis is true, under this hypothesis, most of the data we observe will be here, right? It's more likely to observe something like here. On the other hand, if we do experiments, if we observe some data which is here or maybe even more extreme, then it is the value that under the null hypothesis, the probability is extreme to observe the currently available observation. So that is the p-value. So the vertical coordinates here is the probability density of each outcome computed under the null hypothesis. The p-value is the area under the curve past the observed data point, here the entire green area. So as extreme or even more extreme as the currently observed data points. So that's the p-value. So anyway, there are two main features I would like you to remember. For this null hypothesis testing, go back to here, it means we always assume the null hypothesis is true. We always make a test against a hypothesis of no effect. This red line here, this means null hypothesis. So this is always true. We never calculate the probability of the alternative hypothesis. We do not do that. And the p-value, it is also not the probability that the null hypothesis is true. This is the common mistake, I think. So the p-value is here. I do not feel bothered if I want to repeat again, so I will just repeat again to you because this is important. So p-value, it is the probability of observing results as extreme or more extreme than currently observed, given that the null hypothesis is true, okay? Good. And what we do is to use p-value to make inference. So there's a typo here. So what you can do after today is to watch this video. So it is a very clear and interesting explanation of p-value. Okay, so go ahead and watch it. So related to the p-value, there are two concepts. One is called type 1 error, one is called type 2 error. So here, this table, this is the classic textbook example of type 1 and type 2 error. So I want to skip because there is another illustration which is much nicer. So let's say this is the truth, okay? The truth here says null hypothesis true, there's no effect. The truth here says the null hypothesis is false, there is an effect. So here, this is your statistical test, how your p-value tells you. So the results here says, well, we have to reject null hypothesis. Well, here it says fail to reject null hypothesis, okay? So let's say if the truth is that the null hypothesis is false, this means there is an effect. And then we successfully reject the null hypothesis. This is a good performance. This is a correct outcome. This is a true positive. Relatedly, if the truth is that there is no effect, and then the analysis also fails to reject null hypothesis, this is also a correct outcome. So true, negative. It is negative, and we truly found it. There are some other cases which is a mismatch. There are some errors. So say here, supposedly there is no effect, but the analysis, the result says to reject the null hypothesis, then it is a false positive. It is not positive, but it is a false positive. So this is a type 1 error. So here, the truth is there's no effect. The truth is that this is a male. It can never get pregnant. But then the result says, well, you are pregnant. The opposite is here. The truth is that there is an effect, but then the result says fail to reject the null hypothesis. So even though this lady is pregnant, and then the result says you're not pregnant. So this is the type 2 error, okay? So those are the two type 1 error, type 2 error. You have to keep in mind when you are doing your analysis. Good. A few tests we used very often, and after I introduce the tests, we have the break. So t-test, oops. T-test, one sample t-test. For example, we want to test if the grades of a class is higher than the average of the entire university. When we have one sample, only one group of data. Sometimes we have different group of data, but they are from different groups. So for example, study the IQ performance from the different moods, right? Positive versus neutral. So this is usually appropriate for within subject design. We use independent sample t-test. Another is that if we want to have a within subject design, I ask you questions at the beginning versus at the end of the semester. You are the same person who is doing the test twice, and then because the data is related within yourself, then the corresponding test is a paired t-test, okay? So there is even another scenario where in the independent t-test, there is an equal variance version, and there is also a unequal variance version. But I guess you have talked about this in your statistics seminar. So when there are multiple independent variables, rather than 2 levels, but 3 or maybe even 4, so what we use is a analysis of variance, which is called the ANOVA. I guess you also know it already. So when there are more than 2 groups of or conditions, means more condition means the average to be compared. The most common null hypothesis test is the analysis of a variance, the ANOVA, sorry. So the general idea is to test the ratio between the between group variance versus the within group variance. So here, this is the variance between groups. We use this one divided by this. Same idea, we use this one divided by this. So in these 2 scenarios, the ratio here is larger than here. So here we could expect a significant group difference by using ANOVA. However, we might expect a non-significant group difference in this ANOVA. So recall another example we have in this video watching violent, non-violent, cartoon versus real example. So we could use this ANOVA, we could use a 2 by 2 ANOVA to test if there is a difference or not, okay? Correlation, this is simple, quite straightforward. So this is the degree of relationship between 2 variables, and we often use some index. The most common one is Pearson's R, the correlation coefficient, and this correlation coefficient varies from -1, perfect negative correlation, through 0, no relation, to +1, a positive correlation. So here, positive, negative correlation, some negative correlation, here no correlation, some positive correlation, and a perfect positive correlation, okay? The correlation coefficient R here tells you, and also here the illustration, the strength of the relationship between the 2 variables. And sometimes you are also interested in making predictions. For example, you want to know how much one variable will change on the basis of knowing changes on another variable. Or maybe you want to know how a particular individual with a given score on one variable will score on another variable. So let me give you one example. So suppose here this is age in years, 2 years old, 4 years old until 16 years old. Here it is the number of nations names. So how many nations they can name. Germany, Austria, France, for example, the number of names they could name, the number of countries they can name. So supposedly, as long as they grow up, they might be able to name more countries, right? So assume there is a correlation you first conducted, and you want to also make predictions. And then the way to do it is to run a regression line. So supposedly, if you do in SPSS or in R, we got the regression line. So the number of nations equals to minus 3.15 plus 0.97 times the age. So that means the slope here is 0.97, the intercept is minus 3.15. So this line means every time when we have a 1 unit change in age, so every time there is a 1-year-old increase, the number of nations that can be named, it will be increased by this amount, okay? So this basically means if changing from year 12 to 13, then the number of nations that can be named will change from somewhere here to here by a unit of 0.97, okay? So this is the idea of a simple linear regression. The very last type of analysis before the break is that it's called computational modeling. So this is something maybe you haven't talked about, you haven't heard about during your studies in statistics, but this is very common in decision neuroscience or in decision making in general. The idea is that suppose here this is a participant. We ask participant to make some choice, make some decisions, right? Decision making. We know that the responses, they come from the brain. So we either scan their brain using the machine, the MRI machine, or maybe here we only record the button presses, the behavior. So all of the brain data and the behavioral data, they basically come from the brain from the participants. So what we want to do is that we want to use the data from the participants to inform what might be happening in their brain, right? So as you could imagine, from here to here is not so straightforward. It might be quite challenging to look at because we cannot open the brain to look at what's inside if that's a healthy participant. So what we do instead is we construct a cognitive model or computational model that is trying to approximate what might be happening in the brain. So the idea here is to use a model to approximate the brain, okay? We could also let the model to do this experiment as if this is a person. The model could also generate some behavior responses, like the model will also make decisions from the experiments. So what we can do is we could compare if the participant's choice, the actual choice, match up with the performance from the model, from the data coming from the model. So if here there's a match, we could say, well, the model approximates more or less how the brain works because the outcome here, the observable area, they are similar. And then the brain might work this way. Okay, so this is computational modeling, the general idea. This is increasingly helpful and relevant and important in the field of decision making. So later, when you read papers, you will see many of the studies are using more or less some kind of computational modeling in their research. Good. Any questions? Everything's all right? Comments, feedback, anything you want to share with others that I haven't talked about, but you think that might be important to talk about? No more questions? Yeah, there is a question. It says, how can we be sure that a program will behave as a human would? No, you're never sure. You are never sure. So all the models, so if you hear the news, there are artificial intelligence this and artificial intelligence that, they are somehow only able to resemble one specific aspect of human. So let's say one machine is only able to play the Go game, okay? That could be even outperform human. That's possible. But this Go machine, this AlphaGo, it cannot really help us to make decisions about food choice, for example, what to eat, right? So each model or each program or each artificial intelligence is only able to approximate one very tiny specific aspect of human, and also not even perfect. And this might be a philosophical discussion. So do they have minds? Do they have emotion? This is too much, I think. So we are only talking about the cognitive part. For example, if we ask the participants to learn, maybe we ask participants to do the IQ test, we could also make a program that is also able to do the IQ test. That might be possible that the machine is very able to do the IQ test. Yeah, the follow-up question is, how often is that used in research, or what benefits does it have for research? At least speaking to the field of decision-making, this is used very often, especially after 2000, the year of 2000, maybe 2010, or I guess around 2005, in the decision-making field and also learning and decision-making fields. Paper about this topic, maybe 10,000, maybe even more, a lot, really a lot. Yeah, so the other question, so scientists base the computational models on their theory and then compare the results of the modeling to the results of the neuroimaging to find out if their theory is correct. Yes, more or less, yes. So there is a recent paper, this is kind of challenging in the field of psychology. So some people say, well, unlike physics, psychology sometimes lacks a very well-founded theory. And this is a very challenged idea of the field of psychology. And then some people say using computational modeling is one way to kind of solve this problem, because you know how the model will work. And then you use human data to improve your model, right? You compare if the model somehow behaves quite similarly or differently relative to the human behavior. And then if the human, they behave more or less similar to the model, to the theory you have in your mind, then this theory might be confirmed. Or in another case, if you have a theory, you implement a model, but the human being, they do not perform or behave this way, then this theory is disconfirmed. And then you can use this mismatch between the human behavior and the program behavior to improve your theory. You see this circulation, so you start from somewhere, and then you test if that's account for some interesting findings or not, and then to improve it, and then to eventually find out what is more interesting.